Well, thank you, Dr. Marian, very much for the kind introduction and the privilege to present today. Let me share my screen here, and I hope that you should be able to see that. If so, then we can proceed. It's a pleasure to give this named lecture after my esteemed colleague, Dr. Unger, Roger Unger, who passed away, unfortunately, about 2 years ago, a little more than that, but he was both a role model, a mentor, and a colleague. And I think his contributions in the area of endocrinology, glucagon in particular, are really breaking new paths, basically, in the area of treatment, and now in the area of triple agonists, where glucagon is coming back. He's really, we realize how much he was ahead of his time. But what I want to do today is get you a little closer to this somewhat mysterious cell, the adipocyte, and its impact on cardiovascular disease specifically. It is certainly a cell that most of us have too many of those cells, but we also appreciate that they play a very important role. And if we don't have them, or they're misdistributed in the form of lipodystrophies or lipoatrophies, we appreciate that this leads to very profound systemic pathology. So an understanding of this cell is absolutely essential, even though it is clearly linked in terms of its cell mass with a lot of pathology. You all know maps of individual countries, whether that's Europe, Asia, in this particular case, the US, in this case, a two-dimensional map between obesity, excess adipose tissue, excess number of adipocytes, and in this particular case, type 2 diabetes. And you can see quite clearly, the more purple the map gets, the higher the overlap between the two, making a very strong case that the two are epidemiologically and mechanistically very much linked. And this happens to be type 2 diabetes that we are looking at here. But of course, if you were to focus on cardiovascular disease, there is very much an similar overlap in this particular case, seen here by this map from my colleagues at NORC in the University of Chicago, where you can see that wherever you have high prevalence of obesity, there's a high prevalence of cardiovascular disease. So clearly a strong mechanistic link between the two. It wasn't always like that. Not that long ago, the adipocyte was considered to be a very simple cell. We have it's almost a test tube in the lab, where incoming postprandial free fatty acids and glucose get taken up, esterified, stored in the form of triglycerides, and then released during times of need. Whereas we appreciate now that the adipocyte is a very complex biochemical machine that secretes all kinds of things. Hormonal factors, we like to refer to them as adipokines, lipid signaling molecules in the form of sphingolipids and ceramides, so not necessarily energy generating molecules, but signaling molecules, and then critical metabolites that are highly specifically produced in the adipocyte, one of which, for instance, happens to be uridine, but there will be many others, playing a very important role in systemic metabolism. So we appreciate the complexity of the cell. And in the context of cardiovascular disease, and of course its target organ, the heart, we do know, and many reviews have been written about the fact that there is a inter-organ crosstalk between fat and the heart. And again, the molecules that I just mentioned, adipokines, such as adiponectin, leptin, FGF21, and others, which are either enriched or maybe exclusively expressed in the adipocyte, but minimally enriched. And then the ceramides, potent pro-inflammatory molecules that are tightly linked to insulin resistance, and the matrix, the fibrosis that is characteristic of many diseases in the cardiovascular world specifically. And then, of course, we have in the immediate vicinity to the cardiomyocyte, we have fat pads in the form of pericardial or epicardial fat, and these local interactions in the microenvironment are, of course, also playing a major role in light of the fact that the concentration of some of these factors can actually be very, very high in the local microenvironment. So unquestionably an important role there. And we know that for the overall metabolic well-being, as well as the well-being of the tissues in the immediate neighborhood, the fat stores are essential. They can expand, they can shrink, and the obvious thing, of course, is this is a balance between energy intake and energy expenditure. Energy intake in the form of calorically dense foods, and energy expenditure with a basal metabolic rate, physical activity. And yes, we do have to stay warm. And as such, of course, adaptive thermogenesis is another form of an energy sink. And how we handle excess calories going into the system, or a relative undersupply of calories, very much depends on the metabolic flexibility of the adipose tissue, specifically the adipocyte. And if the adipocyte mishandles this excess energy load, of course, that is the foundation for all evil in the system in terms of metabolic dysregulation, leading to all these other pathological sequelae that we're very much familiar with. So in that sense, metabolism is an area that the fat cells profoundly affect in that area, in that context. But something that we spend less time thinking about is the interaction with tumor cells. We appreciate a very tight link between increased BMI and tumor incidence. And certainly in the US here, the NCI, the National Cancer Institute, along with the NIDDK, really has a major effort at this point, trying to better understand what the underlying mechanistic connections are between excess dysfunctional fat tissue, increased tumor incidence, and enhanced tumor growth. Even in the context of infectious disease, we know that the fat cell is a target for a number of different viral agents, parasitic agents. Just to recall briefly that, for instance, when we have Chagas disease, the causative agent of Chagas disease and the cardiomyopathy, which is associated with it, is a parasite referred to as Trypanosoma cruzi. And this particular parasite absolutely adores the fat cell as a target. It invades it and it sits in there, and it takes advantage of the fact that the adipocyte has a half-life in humans of at least 10 years plus. So this is a wonderful place to reside for a parasite, protected from the immune system, and then ready to break out whenever the immune system is compromised. But I want to bring this mysterious adipocyte cell a little closer to you in terms of the various aspects of it. They start out with precursor cells, of course, like every other tissue, terminally differentiated tissue. And these fat cells, once they're fat cells, the dogma is that they have relatively limited flexibility. As a terminally differentiated cell, they can either increase in size, they can decrease in size, or there are various pathological states under which the adipocyte ceases to exist, and of course can either undergo a low-key exit in the form of apoptosis, or it can necrose and leave a lot of local inflammation behind. More recently, we started to appreciate that the adipocyte does not necessarily have to be a terminally differentiated cell. And in fact, there are conditions under which we've seen that the adipocyte can effectively de-differentiate into a precursor cell from which it can go down a number of different interesting paths. Most strikingly, I think the first observation we made in this context was during lactation. And relatively simple genetic studies, basically, that allow us to paint, in this particular case, all of the existing fat cells in blue, and then stop the painting and watch what happens, for instance, during lactation. You can see that during lactations, the fat cells disappear, milk-producing lobules appear, and we end up with these clearly black C positive fibroblast-like cells. And by definition, these cells actually have to derive from adipocytes. And those are single cell sequenced, revealed to be actual pre-adipocytes. And when lactation stops and involution kicks in, the fat pad regenerates, these cells come back, and you can see that all of the fat cells that come back are still blue, indicating that they actually are still the original fat cells. So there's a full-blown de-differentiation, redifferentiation event for these fat cells. And you can repeat this over several lactations, and the vast majority of the cells remain blue, with a few exceptions here, indicating that that flexibility of the fat cell to de and redifferentiate is inherent in these adipocytes from the mammary gland. So you have this lactation merry-go-around where fat cells come and go. And it's the very same fat cell and pre-adipocyte that circles around here and regenerates the mammary gland post-involution. We also have a very important set of information from the dermal adipocyte, and I want to alert you to the fact that the dermal adipocyte is not equivalent to the subcutaneous fat pad. The dermal adipose layer is a very thin layer, as you can see here. In the dermis, it can expand under certain genetic or nutritional conditions, but it's overall a relatively small but very important fat pad that has the ability to produce antibacterial peptides as a first layer of defense for invasion from the skin. It is involved in hair follicle growth. It is involved in wrinkle formation. So it's a very important fat tissue. And there we make the same observation in the mouse, but it's highly synchronized, a little more complex in humans. But basically, dermal adipocytes come and go and undergo complete cycling, just like in the mammary gland, between fully differentiated cells and precursor cells. Again, single cell sequencing reveals that they're fully de-differentiated and then come back in a synchronized fashion initially. So these are physiological settings under which this happens. In the skin, we can provide an insult to the skin in the form of bleomycin or radiation here. And you can see under those conditions, these adipocytes can undergo de-differentiation as well. But then, rather than just being benign pre-adipocytes, they actually end up going into a myofibroblast-like state, where they produce a lot of the extracellular matrix and the sclerosis that follows suit here in those conditions. And we refer to this process as AMT, as the adipocyte myofibroblast transition. And the more we learn about the flexibility of these terminally differentiated cells, the more complex things get. We do appreciate that the adipocyte has the ability to turn into a myofibroblast. There's quite a bit of data arguing that macrophages can turn into myofibroblasts as well. So there's a lot of inherent flexibility in the system. Now, fibrosis is a very, very important aspect of normal and dysfunctional fat tissue, and I want to belabor these a little bit further in different pathological settings, and then talk about two specific profibrotic molecules that we start to appreciate, leptin and endotrophin. So in the lung, we have fat cell-like cells, lipofibroblasts. They express PPO gamma, adiponectin, and other markers here, and they can actually actively undergo the same process. They can de-differentiate, redifferentiate, but they can also morph into these myofibroblasts. That's something that takes place with relative ease in the context of infections of the lung. We definitely appreciate it in idiopathic pulmonary fibrosis. And of course, COVID-19 and other viral infections result in a similar phenomenon. And we've played in the past around with critical stabilizing factors. The determinant for a terminal fat cell is, of course, PPR gamma, and we have anti-diabetic agents in the form of thiazolidinediones that can actually stabilize this state here, or force a myofibroblast back into a fully differentiated lipofibroblast. And as such, these TZDs have found widespread use in the context of their anti-fibrotic abilities and anti-inflammatory abilities, useful for the liver and definitely validated in many instances for fatty liver disease, but also in the lung, as we can see here. Now, in the lung, more specifically, the fibrosis can kick in massively post-infection, or again, we used here the model system in the form of bleomycin aspiration into the lung, and you stain these mouse lungs here with a trichrome, where you can see the blue stain here is the widespread fibrosis. And over the years, we have appreciated that leptin, the classical leptin factor from adipocytes, obviously plays a major role in the cross-communication between fat tissue and the brain, but there's actually an equally important role for leptin in the periphery, where it can act as a potent pro-inflammatory and pro-fibrotic factor. And you can see here, when we treat here these bleomycin-exposed mice with a neutralizing leptin antibody, there is a dramatic decrease in the fibrosis that prevails in the control mice here with the IgGs, but the leptin here completely reversed this dramatic enhancement of the fibrosis. So unsuspected, but very important, physiologically very important action of leptin action. You can see here how we can both histologically and functionally restore lung function here with these neutralizing antibodies compared to the heavily fibrotic areas in the lungs there. Cancer is another example in the mammary gland, when you have a lesion infiltrating in the context of breast cancer. Similar idea here. So you have a fully mature painted adipocyte. You have these fibroblasts here that clearly derive from the adipocytes as the lesion enters. You can see this both in rodent as well as in human samples. As the lesion infiltrates the fat pad, fat cells get smaller, they delipidate. And again, we have the advantage in the mouse that we can paint these fat cells prior to the invasion of the lesion. They de-differentiate, they can be isolated, single cell sequenced, and actually offer a remarkable spectrum of cell types that these fat cells can turn into: pre-adipocytes, macrophage-like cells, myofibroblasts. And I'm telling you this story with cancer, breast cancer as a background mostly, because that applies to many other settings, including also the cardiovascular setting as well, where we have to appreciate that these fat cells have a lot of flexibility within their system. So what are some of the factors that really drive basically this de-differentiation? And then from that point on, once de-differentiated, what will be the factors that drive these precursors towards a more fibrogenic myofibroblast stage? Endotrophin in this context is a novel quote-unquote adipokine, in the sense that fat cells produce a lot of it, but its expression is not restricted to adipose tissue. It's also a little more complicated in the sense that there's no single gene that encodes endotrophin. Endotrophin is actually a post-translational cleavage product of the mature collagen 6 chain, specifically the C5 domain of the collagen 6 alpha 3 chain, that gets cleaved off and has a life of its own. And this cleavage product here in red, this is actually what we refer to as endotrophin. And we've been working on endotrophin for the better part of the last 12 years. But I think over the last year or so, endotrophin has really entered sort of a new area for a number of reasons, in the sense that it's gotten a lot of attention. And I think I want to bring this to you as well as cardiologists, that this may be a factor that we have to remember in terms of its pathological actions. Everywhere you have fibrosis, particularly in fibrotic adipose tissue here, this is a sample from human breast adipose tissue, stained for endotrophin. You can see that fibrotic fat pads have a dramatic increase in endotrophin signal in this context. That can be pathological in the sense that it can cause an inability to further expand the adipose tissue. It can restrict the flexibility of the adipose tissue. We've looked at this a little bit with my colleague Sam Klein here at WashU, who has an interesting cohort of individuals that are metabolically healthy but obese, or they can be metabolically unhealthy and obese. And we adjust for the same BMI in both cases. In one instance, we have individuals that, despite their obesity, retain insulin sensitivity. In the other case, they lose insulin sensitivity. And most of us fall, of course, into the category of metabolically unhealthy obese. We can measure endotrophin not only locally in the microenvironment, some of it escapes, we can measure it in plasma, and you can see here that the endotrophin levels are actually increased in the metabolically unhealthy obese. And what's even more interesting is that these levels track extremely well with insulin sensitivity and adipose tissue overall function. So more endotrophin in adipose tissue means more fibrosis, more inflammation, more insulin resistance. Certainly an interesting component here clinically, and the same thing can actually be seen preclinically in rodent models. We spent much of the past 10 years defining endotrophin as a driver of malignant tumor growth, of course, starting out again with breast cancer, where tumor lesion is bathed in these adipocytes, very much exposed to high levels of endotrophin, and concluded from these studies that endotrophin would be in many ways a factor that will remind you of TGF-beta as a pro-fibrotic factor. It has some cancer-specific properties. It's a chemoattractant for various cell types. It stimulates this epithelial mesenchymal transition of tumor cells associated with chemoresistance, and it just overall acts as a potent mitogen for tumor cells in this context. Developed neutralizing antibodies against endotrophin. Again, that can be used very effectively in these cancer studies. You can see here a dose-dependent increase of neutralizing endotrophin antibodies, and you appreciate that at higher concentrations where we neutralize most of this endotrophin, the tumor growth is quite significantly reduced under these conditions, highlighting that this is a factor that may have a lot of promise. But we are not restricted to cancer and the oncology space with this one. We really appreciate that this is relevant for any fibrotic disease within the system. Liver fibrosis, of course, the focus of a lot of studies. When we use a preclinical model of liver fibrosis, this is an ER stress model. We call it the MUP-UPA mouse, which Michael Carin popularized at UCSD. You can see that basically with neutralization of endotrophin in this context, we have a dramatic reduction of the pro-fibrotic readouts in the system locally. And with that, of course, we have reduced steatosis, reduced collagen production, and overall improvement in liver functionality when we avoid this molecule endotrophin. Similarly, in the kidney, various models of kidney dysfunction that lead to fibrosis can be used here. In this case, we use a podocyte insult model. And even though this is a very complex slide, the bottom line is it's exactly like in the liver, when we treat this system with neutralizing endotrophin antibodies, we restore normal kidney histology. The fibrosis disappears by gene expression as well as by histology. And upon closer examination, you can actually see that we have a full restoration here of the kidney podocyte function here as well. And as such, is of course also functionally restoring kidney function as such. Now, in the context of cardiovascular disease, endotrophin has entered the field as well. We do see a number of papers that have popped up, initially mostly in the context of HFpEF, heart failure with preserved ejection fraction here, with this work from Bristol Myers and other colleagues at UPenn that looked at intervention. I believe in this particular case, this was SGLT2 inhibitors treatment for HFpEF patients. And they measured endotrophin in circulation prior to initiation of the study. And they actually found that endotrophin is an outstanding marker. If in fact, it's by far the best marker compared to the standard markers for HFpEF. As you can see here, you got the MAGIC score and the classical pro-BNP here. The hazard ratios, as you can see here, for endotrophin elevation in the plasma of these patients is dramatically improved, and it tracks very well with other endpoints, including in this death here, or death or heart failure admissions. So this ends up being a very powerful marker. You can divide, perhaps in a more simplistic way, the patient population into three groups that has either very high, moderate, or lower endotrophin levels. And again, you can see the individuals with lower levels are more protected. High endotrophin spells trouble, and you have a decreased survival probability with the higher levels, as well as some of these other readouts. So it's certainly something which is now emerging as a powerful marker in this particular context, and not just for cardiovascular disease. I think any fibroinflammatory state is associated with an increased presence of endotrophin in circulation. Endotrophin acts locally as a profibrotic factor, obviously. Some of it escapes in circulation. We measure it by ELISA, and you see these quite striking correlations. We know that this is going to be more than just a marker. I think a lot of preclinical data now suggests that we have more than just a marker state here. This is causality. Overexpression of endotrophin in various disease states, including the heart, is actually highly pathological. Neutralization of endotrophin, and I've shown you a little bit of that beforehand, it gives us an opportunity to improve the state of these preclinical models. So very promising overall. Both as a biomarker, I think definitely I put endotrophin in terms of its future impact on par with adiponectin. I believe it's going to be an equally powerful marker and probably lend itself for interventions in both the cancer as well as, more importantly, in the fibrotic world. So we all appreciate that fibrosis is one of the big unmet needs out there. And as such, the neutralization of endotrophin bears great promise in the context of lowering and in some instances even reversing fibrotic states that are out there. But in the context of adipose tissue, I think we have still so much more to learn, and it lends itself really as a discovery target for a lot of new factors that we still need to better define. We usually on our end work with a lot of preclinical models that allow us to hopefully provide interesting clinically relevant models. I mentioned these metabolically healthy, obese individuals that Sam Klein is working on. We're working closely with him because we have the equivalent of a preclinical model for that. We've made on many occasions the world's fattest mice, but what's interesting in this context that these mice, despite being the equivalent of 600-pound individuals, they retain actually their metabolic normal state, their insulin sensitivity, because they very effectively neutralize excess calories into their subcutaneous fat mass and retain insulin sensitivity. So as such, these mice have proven to be extremely insightful in defining what it means to have functional adipose tissue with reduced fibrosis, reduced inflammation. But then we also know that fat tissue is actually far more than just fat cells, which we have to bear in mind. We mentioned these pre-adipocytes, of course, that are very important. We have ways by which we can manipulate these pre-adipocytes metabolically in relatively simple ways. And we appreciate that this is associated with dramatic changes in the overall health of fat tissue as well. We also know that the fat tissue is a reservoir for a large number of immune cells. Certainly macrophages play a major role. And again, these adipose tissue macrophages can be manipulated. Fat pads with altered macrophages can be transferred, and they can convey phenotypes from one rodent to the next by simple transplantation experiments of relatively small amounts of fat. However, I want to spend the final minutes of the talk a little bit about a different model, which is actually a lean, healthy model. We do undergo right now a bit of a revolution in the weight loss field. We appreciate that agents that play along the GLP-1 receptor axis have proven to be very effective insulin sensitizers and weight loss drugs. The next generation that came up and is approved now will be the dual agonists. Those are agonists that act on the GLP-1 receptor axis side along with the GIP, the receptor axis, and activate both of these. And of course, with these interventions, we appreciate that we can get with relative ease and with relatively limited side effects, a weight loss of up to 20, sometimes even 25%. Now, any individual that loses 25% body weight, that means we are dropping a lot of adipose tissue mass. When we lose that much adipose tissue, a lot of things simply resolve. A fatty liver resolves, insulin sensitivity increases. In fact, the dual agonists for the moment are still only approved for diabetes, but of course everybody is jumping on them as weight loss intervention, as a weight loss intervention. So this is something which is going to fundamentally change clinical practice, including in the cardiovascular field. And of course, any number of studies are actively ongoing. I just pulled one here out of the hat with this molecule tirzepatide, that is the approved molecule from Lilly, that is being studied in a large number of different disease settings, including in HFpEF here, actively ongoing, this summit trial, and it will read out next year at some stage, about 12 months down the road from now. And predictably, what I believe we will see that there will be improvements in this setting as well. Remarkable what 20% weight loss can actually do for you in this particular context. But it also begs the question of how exactly do these drugs actually achieve this massive weight loss? And particularly the GIP axis is relatively mysterious still. And we set out a little while ago with my colleague Christine Kuzminski to basically better define whether there's a role for fat tissue as a direct target of these agonists. There is a lot of literature already out there by this, but we really are far from understanding the mechanistic underpinnings of this weight loss. So our approach was to basically study this in fat cells, and we actually studied this by simply overexpressing the GIP receptor in the adipocyte. And of course, in preclinical models, we have the ability to do this with relative ease. We can make mice that just have a lot more of this receptor in the adipocytes, and we can actually do this in such a way that we can titrate and induce the levels of the adipocyte in the adult mouse, so we don't have any developmental issues and have here a fat cell specific GIP receptor overexpressing mouse. And you can see here when you go in different dimensions and label a fat pad with the GIP receptor, we see how the GIP receptor is expressed in these adipocytes. And we can see at the bottom here that this is expressed in a number of different fat pads, the gonadal fat pad, the subcutaneous fat pad. So as such, we can basically equip the fat cell with enhanced expression of this receptor. And when we induce the expression of this receptor, something remarkable happens here. When we put the mice on a high-fat diet, you can see that they gain less weight, quite dramatically so, in fact. But even though they start out with the same body weight, they reduce their weight gain quite significantly. They don't lose weight when we start out with lean mass. What usually happens when we see something like that, and we have a whole bunch of models that kind of behave that way, but we have all kinds of red flags going up. This because it means that maybe we're creating a lipodystrophic mouse model here, which would not be good. But a lipodystrophic mouse model would have a horrible looking liver with steatosis and fibrosis. That's not the case here. So this is an actual model of healthy fat. We can do this with obese mice that start out fat and then induce the receptor. You can see what a dramatic weight loss that brings about, easily minus 30% in body weight. The fat looks great in this context. I mean, there's no pathology in the fat. There's no inflammation. There's no fibrosis. So these are perfectly happy fat cells in this context. Frequently, when a weight loss of that magnitude kicks in, we jump to the conclusion, well, we must be activating brown adipose tissue, which is a little bit less exciting because the therapeutic potential for brown adipose tissue in the clinic remains relatively limited. This is not a browning issue. You can already see from the stains of the fat here that this is not a brown beige adipose tissue phenotype. We see the same weight loss at thermoneutrality. This would basically avoid any kind of beiging and browning, so a same issue there. And what's interesting is when you follow this very acutely as we provide excess receptor, you see that there's a transient massive dip in food intake followed by a normalization of the food intake. And this is quite remarkable because it really is a phenocopy, basically, of what you see clinically with the dual agonist interventions as well. Far more efficient, actually, than just calorically restricting mice. There is an energy expenditure component here as well. So both of these mice eat the same amount. However, the GIP receptor fat is actually much more effective. And again, it does look very much the weight loss clinically, the food intake phenomenon, the energy expenditure is pretty much overlapping with what you would get with these dual agonists. There is definitely a communication axis between the fat to the brain. You can see here when you look at how the brain responds to the stimulation of the fat tissue. It lights up like a Christmas tree by C-phos staining. This is what the neurologists like to use to see brain activity, and you can see how extensive that stimulation is. We activate a lot of different centers that are involved in weight control, energy expenditure, and food intake in this particular context, and we will have years worth of work to figuring out what exactly is happening in the brain. What's important is that there is an increase in energy expenditure when you do this, as measured by many classical means here, without going into too much into the details. Oxygen consumption, lipid oxidation increases in this context. You can take that fat, put it into a seahorse instrument and measure increased oxidative phosphorylation. So that reproduces all of that very nicely as well. But what's particularly intriguing in this context is, and that remains an issue even with the ongoing clinical trials on the dual agonists, both in the mouse as well as in humans, when you stop the drug, you have an immediate bounce back of the body weight. So we don't actually recalibrate the system. We need to have the continued presence of the drug in order to sustain the weight loss. And this is exactly the same in these mice as well here. When you just stimulate for a short period of time and you stop the stimulation, we get an enhanced, immediately rebound back to the original weight. However, if we do this for a more prolonged period of time and stimulate the fat cells with this receptor, we actually seem to recalibrate the system. So we have a metabolic memory instilled on this. This is, for us in endocrinology, extremely exciting, because we haven't really been able under any conditions to actually reestablish this lipostat at a lower level. And as such, of course, this also bears great promise as such. Brief word on the mechanism at the end now. So we agree that there's no Beijing or Browning. The mice don't spend more energy running around, and other cycles, futile cycles of energy wasting are also not taking place here. You throw the kitchen sink, of course, at this, and you walk away with a relatively remarkable new set of observations that involve the sarcolipin circa pathway in the ER as an energy wasting phenomenon, mostly known in muscle so far. This is a futile cycling of the system trying to get calcium into the lumen of the ER, but then just wasting energy by doing so, either by letting it straight out again or by making that translocation highly inefficient. That uses up a lot of energy, and clearly these mice have a remarkable tendency to stay slim over prolonged periods of time. So energy expenditure goes up. So we have an interesting set of observations that can phenocopy the effects of these new dual agonists in the system. As you know, the next generation of triple agonists are already coming up and are in clinical trials. They will take advantage of a combination of the GLP-1 receptor, the GIP receptor, as well as the glucagon receptor action, and promise even higher levels of weight loss. We are using these mice here as a model system to better understand the ability of fat to regulate central effects of metabolism, very powerful effects in this particular context. We don't believe, even though we can phenocopy the dual agonists with GIP receptor overexpression in the adipocyte, we don't believe that this is the sole explanation for the mechanism of action of these dual agonists in the clinic. I know we have a central component to it. We know that there are other site of actions of these dual agonists, but we can certainly effectively phenocopy what these agonists can do clinically. So in summary, I want to leave you with the simple message here that bear in mind that fat cells can play a major role on the basis of being fat cells, but they have the ability to de and trans differentiate into different cell types. Endotrophin and leptin as potent pro-inflammatory and pro-fibrotic factors that have that bear promise in terms of their neutralization to alleviate some of the fibrosis. I told you briefly about manipulating fat cells, stromal cells, macrophages in fat by a simple tool that we didn't get into by manipulating iron levels, but basically pushing these different cell types either down the more glycolytic versus a more oxidative pathway can determine their nature quite dramatically and actually change the overall picture. And then, of course, the GIP receptor overexpression in the fat cell as a powerful illustration of how the fat cells can govern systemic metabolism, energy intake, energy expenditure, strictly from a peripheral perspective, by then signaling to the brain and achieving all these effects. Want to thank my collaborators here, particularly Christine Kuzminski, former postdocs that run their own labs here, and of course some of the colleagues at UT Southwestern that we work with, as well as colleagues in Houston that are an integral part of our joint efforts here. So with that, I would like to open up for questions and stop sharing.